Hello, everyone, and thank you so much for joining us today for the latest webinar in our ALS Research Publication Review Series. My name is Jen DiMartino, and I'm the Executive Director of ALS ONE. ALS ONE is an organization that was founded in 2015 by my dear friend, Kevin Gosnell, who was diagnosed with ALS at age 46 and passed 14 months after his diagnosis. His vision was to advance critical research towards treatments leading to a cure for ALS while simultaneously providing essential equipment, care resources, and an incredible community of strength to individuals living with the disease now. So really to join together ALS research and care. We are based here in Massachusetts. We are really proud to be partnering with NEALS, specifically their NICE Committee. NICE is the acronym for NEALS Information and Community Education Committee. And this initiative is one that is aimed to bridge the gap between the authors of important ALS publications and the ALS community. While amplifying the important work that's happening in the world of ALS, it enables the ALS community to ask questions to gain a better understanding of the article for themselves and for the community at large. So today is an interesting one. We will delve into the publication that is focused on AI and in a specific AI platform. And it's entitled Identification of Therapeutic Targets for Amyotrophic Lateral Sclerosis Using PandaOmics, an AI-enabled biological target discovery platform. It was written by, and apologies if I get these names wrong. It was written by Drs. Frank Pun, Bonnie Heyman Liu, Zi Long, Hoi Wing, Jeffrey Ho Dun Lung, Quinlan Mewborn, Junli Gao, Anastasia Schneiderman, Ivan Ozeroy, Ju Wang, Fen Reng, Alexander Alliper, Eveline Bischoff, Evgeny Ishimenko, Zhoi Mingguan, Qi Zhang, Bei Lu, Jeffrey Rothstein, Merit Sikovich, and Alex Zavarankov. And we are grateful and honored to have one of those authors, the esteemed Jeffrey Rothstein, here with us today, along with an incredible panelist lineup, including Dr. Christina Fournier, who is the an associate professor in neurology at Emory. Dr. Fournier is also a NEALS NICE Committee Co-Chair. Along with Dr. Fournier on the panel are two inspiring gentlemen, Brian Andre and Kevin Robinson, who are both NEALS Research Ambassadors and each living so courageously with ALS. We'll hear from and learn more about each of them soon, but first, it is my privilege to now introduce Dr. Jeffrey Rothstein, who is the John W. Griffin Director for the Brain Science Institute, as well as a professor of neurology and neuroscience, and the founding director of the Robert Packard Center for ALS Research at Johns Hopkins University School of Medicine. Dr. Rothstein was also just recently elected to the National Academy of Medicine. So a big congratulations. It's our honor to have you with us today. And it would be wonderful, Dr. Rothstein, if you'd give us a brief overview on what made you focus on ALS for your career. Obviously your publication, specifically what PandaOmics is, and what drove you to co-write the publication along with your colleagues. After then, we'll then hear from our panelists who will give their own background and connection to ALS before asking questions about your publication.
Thanks, Jennifer. I'm delighted to be with everyone today. Why ALS? So I'm an MD PhD, and the basis of my thesis research, it's not uncommon that those clinician scientists that do basic research or clinical research, when they're done with their neurology training, try to bring that back into the world of faculty practice, academic practice. When I was a resident, there were clues that, resident neurology here at Hopkins, I've been at Hopkins a long time, there were clues about what might be a pathway by which neuron cells in the brain might be dying, including a potential idea about what could be happening in ALS. And I don't want to make this a long conversation on that part, but it had to do with that there were environmental biological targets that caused diseases that sort of represented ALS, not exactly. One's called latherism, another one is a disease that occurs in Guam, where in both places an indigenous population were eating vegetables that contained analogs of the neurotransmitter glutamate. Glutamate makes up everything that allows you to hear and see me and understand what I'm saying. It's a major neurotransmitter in the brain. It was the basis of my thesis. And that work, my early work as a faculty member, which was around something called excitotoxicity, glutamate toxicity, was focused on ALS because of that history. And ultimately, it was the reason, it was the basis for why we have riluzole today. Our drugs, and we'll come back to this, but our drugs that we take and study in patients are almost always based on original research from academics. We don't make drugs in academics for the most part. We rely on companies, but the idea for what should be a drug target, how a drug works, typically comes from academic research. And it was that research I did way back in the early 90s that was the basis for the time Ron Polanco to bring riluzole to ALS. I'm not going to tell you we know today it's by no means the best drug, but it's the most reproducible drug. Until more recently, we have tofersen, which is a powerful. We can talk about drugs. In any event, it was that research that brought me to ALS. And way back in the early 90s, late 80s, we had not a clue about what could be contributing to ALS. That the same applies, of course, to Parkinson's and Alzheimer's. So I was interested heavily from science. The other reason was here at Hopkins, the neuromuscular division, and I think you know that neuromuscular diseases are where ALS fits in, was a very big division. And we are entirely focused on trying to understand the mechanism of disease. So I was mentored by people who really looked at not just diagnosing the disease, but trying to figure out why we have the disease. And ALS was truly an opportunity to carry out that research. So that's way back then. So I'm going to jump now to sort of why this research, which I should make it very clear to the group today, neither Merit Sokovich nor I, we were not original researchers on this project. We were brought in as really consultants. We're authors on the paper, but we didn't do the principal research. Well, to a large extent, we didn't, and I'll dig into that in a second. So I want to be clear. This is a bit of a caveat, a one-off, because I'm going to criticize some of what's in the paper, but we were not the primary researchers of it. The AI approach was really done by this early biotech company in AI. But why? Again, I'm going to come back a step. So having been a researcher for ALS for many years, I'm always interested in what is the mechanism of disease, because mechanism of disease is how you understand and find a drug. Pulling a drug off the shelf and just saying, God, I love niacin and B6. We should just try that. That's typically what I'll call kind of BS research. You have to have a mechanism. You have to understand the disease to come up with an effective therapy. And in all of medicine, that's how we've developed drugs over the years, especially cancer, where we've seen the most effective drugs. So around 2013, 2015, I became quite interested in using a new platform. Almost all of the research that we do in trying to understand ALS doesn't come from patients initially. It comes from using models. I cannot experiment on patients. Way back in the 50s and the 40s and the 30s, that was done commonly. You'd experiment on patients. Yeah, we don't do that anymore in medicine. And I can tell you separate stories about that I've learned from much more senior people. So over the years, we developed tools to find out what is really going on in the disease, because when I see a patient and they're 50 or 60, I don't know what happened to them when they were 40 and 30. I can't, and even when I see them, I can't sample their nervous system. I cannot take a chunk of their brain or spinal cord out, because your brain and spinal cord do not regenerate. Your spinal cord's the width of my finger. If I take a chunk out, you're permanently paralyzed and never can recover from that, in spite of the great hopes of stem cell research, which right now is still very immature. So way back in the 90s, when the first gene was discovered, the SOD1 gene, actually the first tools to study that were based on mice. And those mice were heavily built here at Johns Hopkins. Others developed over the years. And quite frankly, for decades, we used that as a platform to study disease. We learned a ton about the SOD1 mutation from those mice. And for many years, we thought it could be a great platform to find drugs. 20 years later, it's very clear. It's a great mouse. It develops ALS or an ALS-like disease. It's not perfect compared to my patients, but it hasn't been a very good predictor of what happens in patients. And I know the argument, and I've been part of that a mouse is not human. Yeah, we all know that. But how do you study a drug in an intact system that mimics much of the disease? So in many years, we use that mouse. I'm telling you this only because by about 2010, a new platform, a human platform emerged. And that was actually based on stem cells. And back then and now we know that we can take the blood of patients and convert that blood into stem cells, but most importantly, convert those cells back into my patient's spinal cord and brain tissue. And so suddenly we had a new human-based platform. And when I say platform, understand that whether it be a fish, a mouse, a fly, or human cells, they're just model systems. They're not our patients. And we use them as surrogates to better find what could be relevant to patients. It's actually how we discovered the gene therapy for SOD1. That was based on the SOD1 mouse, which is a good mimic for that mutant form of ALS. Probably not a good model, in my opinion. And by the way, everything I'm telling you is my opinion. I'm pretty opinionated, but most agree it turns out not to be this best model for sporadic ALS. But it took years to figure that out. You can look back in time and say, oh, we were stupid. We should have known that. No, we're desperately trying to find drugs. And it was appropriate and still is appropriate for certain conditions to help find drugs. But once we could develop these, and those cells, those stem cells turned into my patient's brain cells, are now called induced pluripotent cells, or IPS cells. It was clear that this could be the next platform. Don't know. Again, 20 years, 25 years after the mouse, some of us throw it out and don't use it as much anymore. The new platform for us to use are these cells called IPS cells. And by the way, before all of this, I hope I'm not jumping around too much. Before we had any of that, of course, we relied on autopsies. And I have a big autopsy bank of tissue from patients who have been gracious enough to have families donate. But that's end-stage disease. I need to know what's going on early, because what happens early is not always the same as the very end of disease. And I say that in part, and again, I'm sorry if I'm jumping a little scientifically, because when someone finally dies of ALS, a lot of bad things have happened to the human body. Those patients haven't been breathing very well, and that changes what's called the acid state of the body. Their nutrition's been terrible at the very last month or 2 weeks before death. So that brain, although it's still very useful, it may not be reflective of what happens when I first see a patient in clinic. So we try to sort of put a patchwork together of the data we learn from autopsy, but for also models. And what happened around 2011, suddenly we had this human, I'll call it now, a biopsy model, living cells. By the way, that became very important to us because that's where great advances were made in cancer. Biopsying cancer, you now have the living tissue. You can study the biological pathways and you can test drugs. I'm telling you this because I'm leading into this particular papers, because based on all of that data, we decided to form a large consortium. That consortium was built out of my Packard Center, but it was backed by a group called Answer ALS, the foundation. It's called the Answer ALS study. It's actually a Packard Center study. That sounds a little egotistical. It's not, but it was really a consortium of 100 investigators and labs around the United States to gather patients, 1,000 patients, study them, so have clinical data, biological data, biofluids, but most importantly, build those IPS cell lines from each one of those patients. So it's equivalent to 1,000 biopsies. And then what you do is you analyze them for every biological pathway you could test. And this testing has been going on way back, this program then finally started around 2016 and essentially ended its enrollment in about 1.5 years. And then the generation of the biological data from the biopsies is only now about to end. So it takes time to do all of that work, but it amounts to about 6 billion data points of biological data from every single one of those ALS patients. It's actually the biggest data set ever generated in ALS anywhere in the world, really. And it was all based on patients. And it was done because we needed really, truly large-scale patient information, both the clinical, but importantly, the biological data. Okay, finally, I'm coming to this. What do you do when you have massive amounts of data like that? You can't analyze that by your average postdoc or young faculty member. You need to integrate. And at the time, this is now about 2020, when we had a fair amount of the data, not all of it, just in the Answer data set. You need to integrate what is called epigenomics, how the environment changes your genes, the genes of patients. Your DNA makes RNA, all the RNA we gather from patients. The RNA make proteins, all the protein data, and the clinical data. So you need to take clinical data as well as all this biological data. How do you calculate things around that? Well, quite frankly, there weren't many people in academics that had those tools. Even now, those tools are still being built. This gets back to this company. This company is one of these AI companies. And to be fair, I don't know them very well. They approached us and said, look, you've got the Answer ALS data. And by the way, you also have, by the way, is the community of all of us as researchers. New York Genome had done about, I don't know, about 100 autopsies and analyzed all of the RNA from the brains of patients, again, at end stage. So we've got this big data from patients' brain, a large data set from the Answer ALS tissues, and a couple other bins of data. And their idea was, could they analyze all of that data? And here's the crux of it. And this is where it becomes very patient-oriented. Not that it ever wasn't, of course it was. Their goal in this study was to not just generate the data, but to identify the biological pathways that the data would teach us. And that's a very plural term, pathways. We'll come back to that and say, if there's a pathway X, is there already a drug that might work in that pathway that we can bring to patients? That was the heart of what their interest was. They were a company because they thought at the end of the day, they might find, oh my God, look, there's 5 different drugs that are existing and they may work on the pathways we discovered here, and they could be useful in trials. And maybe there's some way we can commercialize that. Now, there's a part of that was actually not in our paper. We can come back to that later. But that was the real heart of this. Can we find pathways and assign drugs eventually to those pathways so we might bring drugs faster to patients? I'm going to stop there because that sort of gives you the grand view of me, which is less important today, but why this data is important. Those kinds of data sets, I should end before we talk about this paper in general, those data sets continue to be generated. Largest whole genome, so the DNA data comes from from Europe. It's called Project Mine. Now it's about, I don't actually know the number, but at least 18,000 to 25,000 individual patients' whole genome. In the United States, John Landers at the University of Massachusetts also has a very large data set, about 17 or 18,000 whole genome data sets. That's genome. It's not biology in the patient, but it's their true foundational biology, their genome. We've got Answer ALS. And now we've got some smaller data sets that are groups around the country have generated. Target ALS has a tiny data set of a few hundred patients. ALS TDI has a little bit more genomic data. I should point out a lot of this data, not all of it, is being all centered or put into the Answer ALS data portal now called NeuroMine. That's different from this paper, but buried in all of what I just said to you, when we generate data as academics, because almost all of this data is generated by academics, the whole point of that is to make it available to the world community, not just the US community. And that community has to include companies, just like this paper, which started the process, but because you need everyone to sort of put their finger into the pie to figure out how to use this data, got to be freely available. So the Answer program, when we generated it, patients were consented to make the data freely available to anyone in the world, any researcher. A high school student says, I've got a great idea. Well, this is human data. They've got to have someone over them, some authority figure, but any company and any academic researcher really can gain access to the data. There's important requirements in that, like, so for example, no one can look at the data and try to figure out who the patient was. Yeah, that's illegal. But other than that, it's really available. So I'll stop.
Well, my goodness, thank you. That was a very deep dive into the article and the background on the science behind it. And we are really grateful for all of that information. It's obviously, we know how much goes into writing any article, any publication. So the research, I know that there's, like you said, there's so many different entities that are working within the data space. So I'm sure that there's a lot of collaboration going on, which is really great to see. Thank you so much for that.
Sure.
I'd now like to introduce Brian Andre. Brian, it's truly an honor to have you with us today. I know your wife has been on one of our webinars in the past.
She's right here.
She's right there. Hi, Lori.
We saw her.
if you'd like to go first and give us a little bit of background on you, your journey, before we delve into your questions, that would be really great.
So I was diagnosed in 2016. And I don't know if Dr. Rothstein remembers, but myself and my wife and my sister came to visit him shortly after my diagnosis. And my sister put him on the hot seat answering all sorts of questions. Um, and so I am a very slow progressor. I'm still able to do most everything. I've lost some strength in my arms and legs, but still work out, still get on the tennis court, ride my bike, et cetera, et cetera. My wife really has been really super involved in the ALS world. Several of you probably know her from the work she's done. We were there at the beginning of the formation of I Am ALS here in Chicago with Brian and Sandra. And I've been seeing Merritt as well as Dr. Driss here at Northwestern. And I am on Riluzole and Radicava and a host of supplements by following Dr. Bedlack's advice. And I don't know, that's about all I can tell you, I guess.
Lori was telling us previously on a webinar that it took you quite a long time to figure out the.
Yeah, you know what, for me, I was always very active and athletic, and I started having a little weakness in my upper left arm that I noticed. And I had been seeing a neurologist for something unrelated to ALS for many years. So when I went to the neurologist, they just kind of threw up their hands and said, we'll give you an MRI. It looks like you got a little compression, go see the neurosurgeon. You walk into a neurosurgeon's office, and neurosurgeons do surgery. So they say, hello, let's go have some surgery. And I was like, no, thank you. I did PT for a while. I continued my usual routines. I realized I was losing a little strength in my lower left leg as well. And I went to a physician friend of ours. Actually, she was someone who was running the Shirley Ryan Clinic here in the North Shore. And she looked at me. I think she gave me an EMG, and she told me to go see Dr. Driss. And I got my first diagnosis there. But that was really from the time I first started showing symptoms. It was probably a year and a half before I got that definitive diagnosis. I went for a follow-up diagnosis at Mayo, and then we went on our worldwide tour. We went to see Dr. Rothstein. We went to see Cedars-Sinai, Dr. Lewis, and then we came to Merritt also at MGH, and we had the wherewithal financially, academically, geographically. We were able to do that, and that's something that not everybody is able to do. But we were able to educate ourselves and get to a rather comfortable place after the initial, this is 2016, you have ALS, go get your affairs in order. We went to see everybody and get some more ideas about, what to do and how to carry on, and we've been doing that ever since.
Yeah, you sure have. Your story is remarkable. You and Lori are pillars of strength and inspiration for all of us. Do you want to delve into your questions?
Sure. So I tried to make my questions casual and sort of pedestrian, so everyone in the community might understand and gain something. And so my first question was, I'm not completely clear on how the investigators came to identify the particular gene sites that were investigated.
So on one hand, I can't tell I know how the algorithms work, but what they do is they they took, as best I recall, they took different bins of data. So they took the autopsy data, they analyzed that. And there are algorithms already independently that can say, put in all your RNA data and what pathways look to be the most altered. Usually that means which genes, or in this case, which species, meaning an RNA or a protein, which ones are the most increased or the most decreased. And these algorithms, and these are well known for RNA and proteins, begin to tell you what pathway. So if the pathway to use glucose, this is not necessarily the case for ALS, but just in general, to use a nutrient like glucose. If there's 5 different genes involved in that and 4 of them are altered in these analytics, that's going to tell you that pathway is somehow implicated. And that's what they do. There's a pre-existing data. They don't generate new data. They take pre-existing biological data, which I should point out, which is why these kinds of analytics are going to change over the years as we learn more and more basic biology about how proteins and RNAs and membranes and cells interact with one another. But when they do these analysis of the different bins of data, the motor neuron data from the iPS cells, the transcriptome data from the brain tissue, as 2 examples, then they get those pathways, and then they begin to come up with, well, here are all the pathways. This is where the guesswork comes in. And this is actually why they did that fly experiment, which one of you may ask about later, because now you get pathways. It's never just one answer. None of these ever give you one answer. They may give you 20 answers. And so in their case, because they weren't really cell biologists, we often give hierarchy to pathways. We say, that one makes more sense. I'm going to give you an example in just a second. And then they looked at them and said, well, this pathway, it's involved in glucose. What drugs might improve that? Or what drugs might inhibit that? Because there's too much of something that's already known to be bad for the body. And I'm speaking in generalities, but that's actually how these programs generally work. But then they would compare, was the pathway found in the living biopsy, the motor neurons, the same as in the brain? If they were the same, that gives them confidence that that's probably relevant to something that's going wrong in the disease. But again, when you look at the tables in those papers, there's not one, there really are many. Now I'm going to tell you an aside why that's important to have people eventually test those ideas or bring in outside knowledge, which one of you in prior questions brought in the concept of bias. Because whenever you look at those pathways, you bring in an inherent bias of what I think is important, but I may not be right. And the point of AI sometimes is to be unbiased. It's not who's right. What does the data teach us? We actually have in the whole Answer ALS program, just for the iPS cells, we had an early AI team from MIT, Massachusetts Institute of Technology. These are young guys, men and women, graduate students who can really do those equations really well. And they started looking at the data and they said, well, here's a pathway that looks really interesting. It's the collagen, the protein that's made up in your fingernails. And we said, that's interesting. That's bullshit. Like, who cares? Because there are no fingernails in the brain. And fingernails have nothing to do with ALS, meaning you get a lot of hits from these. Hits are the output, but you have to filter it. And often the way, and I may be jumping ahead to other questions, but the way you often filter it is to say, okay, can I test that idea? You go back to a living system and say, yeah, this is important. This gene does make sense. And that's why they brought in, actually at the time, a postdoc of mine, Ke Zhang, he's one of the authors, and said, you know how to use flies. We can test pathways really quickly in flies. Is this pathway really injurious to cells? And will a drug or fixing that gene fix it? It doesn't mean that that's the drug for a patient. And by the way, a fly we know is not a person, but it's a very quick way of testing biologically, is that pathway dysfunctional? In flies, they often use their eyes. The eyes degenerate really quickly. If you look at those pictures, those are their eyes. And an eye is made up of many little, we'll call them eyeballs in a fly. It's not really that. And they look for degeneration because it's a very common tool. Is it reflective of the real pathway in humans? Maybe sometimes, but also maybe sometimes not. So I'll stop there. So I hope that gives you some idea, very crude idea of what we try to do with the data and choose pathways, but it's still always a guess. Then, and even now, years later, using AI, it's still a guess as to what's the most important pathway. At some point, you have to test them. And when you test them, you line them up and say, that one makes sense or that one doesn't make sense. There's still an element of human testing that follows those analytics.
Okay, thank you. Am I going to ask my second question or are we going to move on to somebody else?
I think in the interest of time, let's see, it's, yeah, maybe what we do is, we can pivot to the next, person. Dr. Fournier, it's an honor to have you back with us today. You do so much for the ALS community. Do you want to, ask your questions now and give us a little bit of background on, all that you do in the world of ALS?
Sure. So I'm Christina Fournier. I'm a neurologist in Atlanta at the Emory ALS Center and the Atlanta VA, and a clinical researcher. And excited about this topic. I was kind of a math major and math nerd in my former life, and I think that AI is a tool in ALS is really exciting. And I know it scares some people, but I think we need to be kind of early adopters and figure out how to best use this. And particularly in a disease where there's so many factors we don't understand and there's so many contributors, I think AI is a really cool tool to have. So excited to delve into this today. So Dr. Rothstein, I think so we had some people look at this article and were a bit overwhelmed by it. It's a lot if you're not used to kind of AI. And so I wonder if we can just kind of make sure we do the Cliff's Notes versions for others that might be overwhelmed. So can we first start with what kinds of data went into these models, and how? Yeah, let's start there. What kinds of data were these models even looking at to kind of make predictions?
Yes, I'm pulling up in front of me because I don't remember either. So it's actually what I really said earlier. They used the data from human brains, so RNA from human brains. This is done at it's the New York Genome. It's a consortium of, I don't want to waste time on descriptions, but it's a consortium of many sites that contributed brains. They took the big answer ALS data set that I mentioned to you as well. They also took little bits of data, some clinical data as well. I don't know honestly how they ever were able to use clinical data, but the big data sets were really transcriptome from Answer ALS and the Target ALS, not Target, sorry, the New York Genome, whole genome data sets. And then I'm looking to see anything else that they had here. Yeah, it was mostly those two data sets were used.
Okay, so these are like
autopsy and living cells, think of it that way.
Okay, so these are like huge data sets with like lots of genomic data, lots of RNA data, something that you and I would look at, and it means nothing to us because it's like an overwhelming amount of data, right? Okay.
Absolutely.
And so you put all of this into these models, and they put some filters on them too, I believe, right? So it was interesting. They could look at things like where grant funding, and I thought some of these filters were really interesting. And I think they kind of played around with them to kind of see what would come out of the model. Do you remember that?
Yeah,
part?
I don't remember, but I actually looked back because I said years ago. Yeah, they were kind of strange filters, and to be fair, I'm not. So, for example, Christina just mentioned they use grant funding, and somehow they could have dug up like scores on a grant. What that really tells you is they're sort of saying, well, how good was existing data, and how did peer reviews, how did other physicians, scientists, or scientists think of that data analytics? Interesting way of looking at things, but they included that too. I thought that was funny. I tend to think of what's important in data is what's the real data and what's the existing publications. The reason that's important, Christine mentions, because once you have all the real data, then you say, well, what's known in the literature to tell me what the data means? Well, one of you mentioned garbage in, garbage out as a pre-discussion. When papers are published, there's peer review, but it doesn't mean that every paper published is of high quality. It's published and everyone assumes the high quality, but once you're really in the field, you sort of might say, yeah, that was a big paper, but it's actually really shitty data, quite frankly. And companies don't know how to filter that. We say that because when we began the Answer ALS program, we met with IBM Watson to begin to say, well, how are you going to analyze our data? Because they were one of the early AI analytics that we thought about bringing in. And their idea was just to do that. Let's take all the papers and what we get from them. Okay, that's not an entirely perfect filter. Or maybe it is because it's unbiased. I don't know. And they did some of that. I don't know how they did it, but they mentioned that. But there's no way of knowing how they did that from reading their paper. So I hope that answers your question about my lack of knowledge.
Fair. No, and that's the thing important. So the good and the bad are that these models are, there doesn't require a hypothesis, right? Like it takes everything in, it looks for these patterns and associations, but it's not based on, I think that this is the important pathway or this is the important gene, which I guess is good and bad, right? Because like you said, you might have something with a lot of with an association that's not cause and effect, or you might find something new that we're not able to see. So I guess that's but it's good from a hypothesis generating standpoint, right? Because you can get all of these.
Yeah. Yes.
Okay.
I hope we have time. So I'm going to give you, I don't want to babble too much, but I'll give you a great example of that. For the
Okay.
last decade, once C9 gene was discovered in ALS, there were two important theories. One had to do with RNA made from that gene, and the other had to do with these polypeptide proteins made from the gene. And there are 5 of them. And over the ensuing 10 years, there were very high profile papers. To us, high profile means like the top journals on how these different polypeptides can actually cause disease in mice and cultures and fly. Actually start with flies. This gets back to one of you dinging flies. And quite frankly, you were right, because that drove eventually companies to try to lower some of those proteins. And we did in the clinical trial. Complete failure. And yet we dramatically lowered those proteins. And that said that, yeah, they were really nice papers, really high profile, and god darn it, that didn't tell us all that we needed to know. And in fact, what happened in the field was we were driven, there were like 2 roads to go down, and we were driven down the road on the right, and we actually didn't do as much research on the road on the left. And now today, we think years later, we should have spent more time looking at the research on the left side of the road, if you follow my analogy there. And so we can be driven by high quality science, leaders and all of that doing that, and be mistaken. Time eventually sorts that out. It's not time that my patients have, but it is the natural evolution of science sometimes. Sorry.
No worries. So, okay, so I'm going to go back to kind of the cliff notes of the paper. So we put in all this data, some filters, they messed around with it. They also put in kind of drugability, right? So for prioritizing pathways that we have a drug or some way to affect that pathway, right? And then they came up with a list of potential targets. So then can you walk us through how they use the fly model to then test some of those targets?
Great question.
Okay.
So now they have, let's just say 20 targets. And it turns out in a fly, and the targets meaning it's a pathway and there's a gene involved in the pathway. And with flies, unlike mouse or any other model system, there's like a gazillion gene mutations in flies. And you can say, oh, that protein X, there's a fly version of that. Let's take the damaged version and see if it really means something in a fly that gets ID generation, and can we fix it? So they used a living animal, that's really an insect, not an animal, to test, is that pathway somehow relevant to why cells die? That doesn't mean it's important in patients, but it was a quick way of testing relevance. And I should point out that's a bias of science reviewers. They say, great, you studied this artificially, but how do we know it's relevant? Let's test a living system. You can't test 20 different targets in a mouse quickly, but you can do it in a fly in a matter of weeks. And that's what this young investigator did, Kozang, and he tested them. It doesn't mean they're right, but he tested them. And then having that data, they could say, this is going to come to your upcoming question. Okay, are there then drugs for those pathways?
Okay, so you're saying the fly is kind of a good genetic screen because it's quick and you can get through multiple candidates, but you're certainly not going to go from the fly to a treatment in people because it's other steps need to happen between that.
But it does happen sometimes, and if you had enough time, I'd tell you about that too.
But I guess generally the advantage is just the ability to screen multiple things in a quicker fashion.
Yes.
Okay, very good. Well, I think I will hand it off to Kevin from here. Thank you. And I believe Kevin is going to be using his speech to communicate. So I don't know if one of us needs to read his questions or if it'll be text to speech.
I think he's going to use text to speak. Kevin, while you're teeing it up, I just want to say it's an honor to have you here.
I'm Kevin Robinson. I'm 63 years old. I am a retired Navy officer, having served 41 years on active duty upon retiring from the Navy in June 2020. 6 months later, in January 2021, my symptoms began with slurred speech fasciculations, jaw tightness, and uncontrolled yawning. Because I am a salivary gland cancer survivor, the initial suspicion was a return of neck cancer. I was diagnosed with bulbar onset ALS in July 2021.
chart for discovery and development and shows patients/ALS goals being involved in providing feedback on proposed targets to select the best candidates for further evaluation. Question: what expertise, if any, do patients bring to the table in selecting targets for further evaluation? Put another way, is patient feedback on drug development actual value added at this stage of the process?
All right, so let me go to the first part of that and remind me to go back. First, I saw that in the paper. I got to tell you, I don't remember any patients are involved in any of this discussion. So they may have added, I don't know. All I know is I don't think Merritt and I recall any patients involved in any of this particular paper. So it's an idea. And then the question, is it a good idea or good is not the right choice of words. Is it a valuable idea in choosing pathways? I don't know. If I'm honest, I don't know that answer. I mean, one can easily say if we're being, let's call it humane, politically correct, sure. But is there a scientific rationale to bring someone who does not have background in biological pathways to make a decision about what pathway to pursue? On one hand, I can say, yeah, I'm not entirely sure. On the other hand, it could be, well, what if there were 5 different pathways and one requires a very toxic drug or not so toxic drug? Do you then add patients in a decision tree on that? And I would say, yeah, probably. Physicians sometimes tend to be, and I'm not going to say I'm that way, but in general, we can be a little overprotective about that. Oh my God, you shouldn't take that drug. It's going to be toxic. I say that in part because sometimes I've had patients who will come to me and seen other physicians, often not ALS neurologists, and said, oh, I'm not going to put you on that drug. It doesn't work. Or I'm worried about toxicity. And I'm thinking, okay, you don't have the disease. That's a patient decision. Educate people, let them know about the full range, let them decide. And so in that sense, I do believe there can be value. Do I think a patient who does no background in the biochemistry, molecular biology of a pathway can make an appropriate decision? That I'm not quite sure of. Again, you could educate them, you could take the time to educate them, but I think the other side of that is if you have multiple choices, then it's time to bring a patient's voice into that and not be so biased as sometimes patients can be. And by the way, there's another part of that where companies can be concerned about, litigious things. Oh my God, if we do that, there'll be, follow-up suits because of toxicity, all those kinds of things that come into play that interfere sometimes with what we do with patients or what companies do. So I'm breaking it down into 2 bins. You may not like that, but I'm just sort of being honest about the way I think about how decisions are made about science. I don't know if Christine wants to add her thoughts to that.
No, it's a good point. I mean, I think certainly we all value and think it's important to have patient feedback in anything that involves people living with ALS in terms of clinical trials or treatment priorities, that sort of thing. But it is a fair point that if you're just kind of looking at mechanisms and which genes are important, how we would incorporate lived experience into that, I'm not really sure. I'm not even sure either. So I think it would be interesting to know what that, because it is in the paper, kind of what that things actually involved. Because it wasn't intuitive to me either. So I think that might be I think that sort of thing.
Yeah, I honestly do not know where that came from. And you might say, geez, if you were an author, why didn't you catch that? And I didn't. It was a long time ago. I can argue ignorance or whatever. Yeah. Now, to be fair, that's not to say patients don't write to probably all of us at times and sort of say, I've read this and I've read that and I've woven my own theory about what's going on. I get those emails periodically, and that's great there, but they're often not fully informed about the full range of what science going on. And that just happens sometimes. And if you have the time, you educate. This happens in clinic all the time. Almost every week, someone will come up with, I read this, I read that. What do you think about that? And that's my job, to educate and also to listen. And part of that listening, though, is education about what I know, what you know, and do we come to a consensus on how to interpret things. To be fair, I love those discussions. This is where and all of you know as patients, this is where the hour visit just becomes so annoying because they're really kind of fun. I mean, at some point, and I can't speak for Christina, but I have, and I know Emory does, a multidisciplinary clinic. Yeah, they handle a lot of things. My job is, in a way, it's that education part. What can I teach you? What can I help you with? What can I unravel? Yeah, someone else knows a better neck brace than me. Why should I make stupid decisions? That may be me. Maybe I'm just stupid at some of those things. But and so those discussions are really great. They're tiring, especially in a day, but
Yeah, we get really good, insightful questions for sure.
Yeah.
I wonder if this use of AI is both an underutilization of the potential as well as a primrose path in ALS. I pose that thought based on the observation that repurposed drugs are primarily useless in ALS.
I don't know that anyone's taken this data from this paper and then used it to do something else. I didn't do a reference check afterwards, as an example. The fact that they did it for the first time, I think is important. It says you can do that. They were focused, as you pointed out, on repurposed drugs. I think other companies will care less about, well, companies almost never care about repurposed drugs. They can't do anything with them. They need to be a better drug. And by the way, that's why often repurposed drugs, I think you're quoting my private comment to you, are useless because they're never optimized for the use. You know, a good example of that, they're botanicals that are used, echinacea, you know, in your cereal. You know, people think, oh, they're really good. Okay, they're not the right dose, they're not the right potency. And so if you have a clue from a repurposed drug, that's interesting. If you want an effective drug, you usually have to make it a better drug, meaning much more potent in efficacy. And the reason privately I mentioned that nutritionals are repurposed drugs are often useless is because I've been doing ALS trials since the early 90s, and there've been a lot of repurposed drugs. In a way, just like this, someone did something in the SOD1 mouse, they've all been failures. They're not potent drugs. And the reason that's important to really acknowledge is tofersen. Yes, tofersen won't apply to the vast majority of ALS patients. You know exactly what the cause of disease. You built the best strong molecule for it, and look at the effects. Years later now, 50% of patients have a clear improvement in something we never even conceived could occur in ALS by turning off that gene with a powerful, very well-targeted drug. And I'm going to tell you, I think the same thing will happen with the FUS gene therapy. There are other pathways that we know about in ALS that are early, meaning early in the event of the disease. One is called TDP-43, and I deeply believe those are going to be powerful targets. That doesn't mean that drugs that are used at other end of the spectrum, anti-inflammatory drugs, the biggest target in ALS, won't have some efficacy, but I strongly suspect they'll never compare to the potency of these really well-targeted drugs. My opinion, and I'll do trials on all of them unless the company's unethical. There are a few. So is it misleading? I don't think it's misleading. It gives clues to what someone can do. And that gets back to when you said it's misleading. This is partly why Merritt and I really agree that putting drugs into this paper would have been a mistake.
Target in this manuscript, given Trehalix was identified as targeting PP3CB several years before publishing this manuscript. What makes it novel?
I don't know that answer, to be honest. I think you wrote that to me. I honestly don't know. Novel, one could argue if someone published something earlier and then it's brought up again, you shouldn't use the word novel. You should say, you should use a different way to describe it. Or it could be that the authors just didn't know about prior publications. I don't really know. Sometimes a pathway, so it's not uncommon when a pathway is discovered. Yeah, we know about it, but if you've now found a drug for that pathway, we use the term novel because no one drugged it before. Again, I may not be answering your question directly, but that kind of nomenclature is used often. You know, C9 was an interesting mutation until we had a drug. The drugs for C9 became novel when we had a drug for C9, even though it was not a novel discovery about the pathway. If it's not the best answer to you, but it's an answer.
That's great. Kevin, thank you so much. Your questions were really, everyone's questions were really incredible. And we thank you, Dr. Rothstein. This has really been so very helpful, helping us to better understand and learn more about PandaOmics, AI as it relates to science and data sets. But most importantly, why we're here is to take a deep dive into your important article. To my fellow panelists, I can't thank you all enough. It's been a really exciting way to learn. Dr. Rothstein, I've asked our past webinars, the authors, what is the biggest takeaway from your publication and from today's discussion that you hope the community will gain?
Yeah, it's a great question. I think the most important thing is, and Christina mentioned this, which is that it might be the first step in a publication, but it's the highlights the direction that many of us think we not just want to know about, that we almost have to go into if we're going to find additional targets for ALS therapeutics. And so I think, even though I'm not going to tell you this is an A plus paper, I'm going to tell you it is that first baby step towards what we need to do and to educate our community about that. And the only other thing I'll mention, and I mentioned earlier, keep in mind that we don't know what's the best way to do AI analytics, and that will grow over time as to what is the most important approach. We know how to evaluate science in other ways. This is new to us, and we're going to have to learn how to do that properly.
Mm-hmm. Well, that's terrific. Again, thank you all so much. I will say that NEALS and the NICE Committee will be holding a live upcoming webinar on just an overarching topic of AI over the next month or so. So stay tuned for that. We also have an additional webinar that we're producing with them on AI and another publication. So definitely stay tuned for some great upcoming episodes. And thank you all so very much. I wish you all the best, and I have great gratitude for each of you.
Thank you.
Thank you.
Thank you. Thank you, everybody.